want to start the presentation by thanking the organizers, Neil, Daniel, Ivan, and Cedric, for inviting me here today, also for the very kind presentation of Daniel. I'm going to be talking today about the visualization of chromosome organization and transcription in single cells. So I think I don't have to introduce this very much in this audience. You all know that DNA is a very long molecule. This molecule, despite its length, has to be compacted in the very small volume of the nucleus. And we have known for a very long time that despite this very large degree of compaction, chromosomes can be highly organized. And so this has been observed probably for the first time by Walter Fleming in the 1980s. And he made a seminal, beautiful drawings of mitotic chromosomes. So since these first drawings of how chromosomes are organized during mitosis, it required more than 5 decades, and the discovery or the development of a new technology, X-ray crystallography, in order to resolve the atomic structure of DNA. And then 50 years later, to realize how DNA is wrapped around histone particles in order to form nucleosomes. More than 10 years after this discovery, developments in microscopy and leveling technologies allow people to realize that chromosomes are organized in discrete territories, also in interface cells. And so in these mesoscales between nucleosomes and chromosome territories, there have been many models for the last, I would say, 40 to 50 years, but many of them have been very controversial. And it was not really up to until the advent or the development of a new technology, which is high-throughput sequencing, that would now have a new view of the levels of organizations between nucleosomes and territories. So there have been many different technologies that were empowered by high-throughput sequencing. I'm not going to go through all of them. I think you have the opportunity during the workshop to be properly introduced to many of these technologies. So I'm just going to say a few words about 3C, or chromosome conformation capture. So this technology relies on the cross-linking of protein DNA complexes, so that from time to time, you are able to capture interactions between two loci of DNA that may be very far away in the sequence space, but happen to be very close by in space, in physical space. So this, combined with high-throughput sequencing, enables us to now draw maps of interaction. These maps of interaction, or contact matrices, you will see in this presentation, and I think the following presentations in the workshop, are formed by essentially looking at the chromosomal, so the x-axis here on the top, and the y-axis represent the genomic coordinates, and the color map represents strength of interaction between any two loci in the chromosome. So the first notable feature that you see here is this strong diagonal from the top left to the bottom right. And this just represents the fact that DNA is inherently a polymer, right? And so any two loci that are close in sequence will also interact physically more often. But the important discovery that is shown in this map is the presence of these squares on the main diagonal. So these squares represent regions with higher levels of interaction and were called topologically associated domains. And so there is several important features of TATs. Probably the most important one is the presence of domain borders, which defines the TAT itself. And so if we take the contact map that I've shown you before, you rotate it 45 degrees, and you remove the bottom part, you can see these upright triangles. And so in these upright triangles, you can see clearly the border. It's shown here by 2 arrows. And people have seen that the borders tend to be occupied by housekeeping genes, by active polymerases, active epigenetic marks, and most notably chromatin insulators, for example, CTCF in mammals. A few years ago, after the improvement in resolution of high-C and 3C technologies, people were able to now visualize the specific interactions between 2 different loci. So these specific interactions can be shown in the map as a spot, a bright dot or a spot that is encircled here in the circle in the drawing. And in this particular example, this is essentially telling us that the two borders of the stat are interacting together to form a loop, right? And so this gives rise to one class of stats that's called a loop domain. So a domain is a tab where the two borders are interacting together specifically. And so, of course, since the discovery of stats, people have tried to see if these stats have or not a functional role. And so since very early, people have realized that stats are conserved among species, that the internal structure of stats changes between different cell types. And by doing genetic manipulation, people have been able to show that removal of borders leads to ectopic expression and to developmental defect. In addition, if we look at how genes and regulatory elements of gene expression, such as enhancers and promoters, are organized in stats, we see that most of the time stats tend to encapsulate these elements, right? And so this gave rise to the hypothesis whereby the genes and the regulatory elements of the genes are all encapsulated in the stat, and so the enhancers can more readily contact the promoters, the correct promoters, and of course, the enhancers that are supposed to be activating this gene in this stat number 1 cannot activate wrongly the gene in the neighboring stat. And so all of this evidence supported the idea that stats may play a role in the functional organization of the genome. However, in the last few years, there have been many other evidences that suggested that this may not be the case. So first, people have been able to show that the loss of stat insulation leads often to very small effects in global transcription. Also, the fusion of neighboring stats does not often change the gene expression of the genes within the stats. And stat rearrangements, very large stat rearrangements, again lead to small changes in overall gene expression patterns. And very recently, people have also shown that enhancers can indeed communicate across stat boundaries. So all of this evidence put into question the role of stats in transcription regulation. And so the main question that we wanted to answer for a while was whether stat structure changes after transcriptional activation. And of course, the idea of this would be to try to shine light into the roles of stats in transcriptional regulation. And so I've told you in the last previous slides that ensemble sequencing technologies were very important. They were able to discover or reveal and characterize a new level of chromosome organization. However, they have some disadvantages. For example, most of these technologies, ensemble sequencing technologies, are unable to dissect single cells. And what I mean by this is the following. Imagine that you have a sample, such as an embryo, with different cells. Each cell here is a circle. And each of the cells displays a different chromosomal organization, right? And so these different organizations is represented here by different gray colors. If you cannot dissect heterogeneity, that means that when you perform an ensemble experiment, what you are going to get is ensemble information of the average contact frequency between any two loci. And so you're not going to be able to dissect the different organization of these cells. So why is this important? Imagine that I tell you that a subset of these cells are expressing a specific gene, and the expression of this gene leads to a different chromosome organization to the cells that are not expressing this gene. Because we cannot dissect heterogeneity, and because we cannot, of course, maintain spatial information, we don't know where our contacts that we're getting are coming from in the sample. That means that the only thing we get is the average contact frequency and the average transcriptional information. And so this means that it can be very hard to actually correlate 3D structure and transcription state just by relying on ensemble sequencing technologies. So for a while, we wondered whether it would be possible to visualize the organization of stats and the output of transcription simultaneously in single cells. So to be able to do this, we need several things. First, we need to be able to image single cells with spatial resolution. We also need to be able to resolve chromosome structure. And of course, we need to be able to simultaneously detect the transcription output. And so we started this journey a few years ago, and our first very naive in hindsight experiment was to label all of the repressive domains in Drosophila cells and visualize them using conventional microscopies. And this is the kind of picture that you get. So you see a number of blobs of different sizes. If I zoom into these blobs, what you can see is essentially that we don't detect very small domains, very small foci. We just detect the very big ones, and all of the big ones even tend to have the same circular shape. The reason for this is that conventional microscopies are limited by diffraction in the maximum resolution that you can achieve. That means that very small domains you cannot observe because of the contrast, and very large domains, they all look spherical, even though they may have an internal shape. And so thanks to the development of several technologies over the past 2 decades, these technologies are globally called super-resolution microscopies, and they have allowed us now to beat the diffraction limit of light. I'm just going to talk today about 2 technologies. One of them is 3D Sim, that enables for a 2-fold increase in resolution, and the other one is Storm or Palm, that enables for a 10-fold increase in resolution. So what do these increases in resolution really mean, practically speaking? So imagine that I show you this confocal image. You may be thinking, well, I see a green background here, and maybe a blob at center. If we increase the resolution 2-fold, now we can maybe see that this blob at the center is made out of 2 blobs, and maybe you can even distinguish that these 2 blobs are sort of linked. And it's only when we increase the resolution by 10-fold that we see that this is actually a picture of the hand of God. This is Maradona striking a goal against England in 1986. You can see now that these 2 blobs were actually the 2 different players, and that the hand of the goalkeeper was actually the linker between these 2. Right? So can we really use this power of increasing resolution in order to be able to visualize epigenetic domains? So of course, we did this experiment. We did label cells the same way as before, but now we use the Storm that increases the resolution by 10-fold. And now what we can see is, of course, different domains. We can see very small domains, and we can see also very large domains, and we can distinguish the shapes, very regular shapes of very large domains. Of course, we can do a similar experiment by labeling active marks. This is K4 methylation 3. You can still see domains. What you see as a main difference is that the active marks tend to be much smaller than the repressive marks. And this makes sense if you know that most of the time repressive marks tend to be much larger, genomic speaking, than active marks. So this first development allows us to have spatial resolution in single cells. However, it has a main limitation. And the main limitation is that while we can resolve epigenetic domains, we don't have the genomic specificity to be able to tell which domain is which, right? So I cannot tell you here, for example, which of these is tab number 1. And so in order to solve this problem, we have turned to the use of oligopane fish. Oligopane fish is a technology that relies on the use of dozens of very short oligonucleotides that will intercalate into the DNA, so they can be used to label very large regions of DNA, and at the same time very small regions of DNA, so they're very flexible. They have been developed by Brian Vellibo at the lab of Tim Gu in Harvard about 10 years ago. And so in our first experiment, what we did, this was a collaboration with the lab of Giacomo Cavalli, and these are experiments performed by Quentin Sabo, a postdoc, PhD student in his lab. And so what Quentin did was to produce an oligopane library that targets specifically this tab number 1, you see here in pink. And the second library that targets tab number 2, that you see there on the right-hand side. So when you visualize this using super-resolution microscopy, in this case was 3D Sim, what you observe is 2 different volumes. The increase in resolution allows us to visualize these 2 volumes separately, and the specificity provided by the oligopane fish allows us to now know that this purple one corresponds to tab 1, and the cyan one corresponds to tab 2. Right? The main biological result here was that these 2 volumes or 2 domains appear segregated in most of the cells. And so this technology, of course, now allows us to maintain spatial resolution and give us specificity labeling However, I want to say already that the labeling specificity afforded by this is, however, very limited in the sense that I can tell you here that this is a volume occupied by tab number 1, but I cannot tell you where the function elements, enhancers, promoters, insulators are located within this tab. And so in order to be able to measure the interactions between function elements, we have implemented another technology where we also rely on oligopanes. The main difference with the previous method is that the libraries are very small. Instead of just labeling the whole tab, what we're labeling is exactly a function element. And so we do a similar experiment. We visualize the location of this spot using Supersolution microscopy in 2 colors. Each locus then appears as a blob of a different color. We can localize the center of this spot with very high resolution, and then we can measure its 3D distance. Of course, then we can establish a criterion to say whether these 2 loci are interacting or not. And so what we say is that if they are closer to a certain distance, then they are contacting each other. If they are far away, they are not contacting each other. And of course, the question is how to establish this distance. And the way we've done it was to label twice the same locus in 2 different colors, right? There, the colocalization should be 100%. And so then we can tune the value so that in all of the cells that we observe, we get 100% colocalization. So this threshold, this diameter of this circle tends to be of the order of 100 to 150 nanometers. So if we use this method to now calculate contact frequencies from imaging data, we can measure these contact frequencies in a locus. This is a region that contains 3 different tabs, and each of these arcs represents interactions between 2 loci. You see straight away that there is more interactions inside tabs than between neighboring tabs. This is what we expected. And so this is a great technique in order to measure contacts between different elements. However, we can only observe 2 elements at any given time, right? So to summarize what I have shown you so far, we have implemented STORM in order to be able to visualize epigenetic domains. But in this case, we did not really have enough genomic specificity to be able to tell you which is domain 1 and domain 2. We have then combined Supersolution with oligopanes. Now we get enough specificity to be able to visualize single tabs. However, we cannot say where the function elements are located within this volume. Finally, we have developed a technology where we can measure pairwise distances, but only one pairwise distance in any given cell at any given time. And so what would be the holy grail? The holy grail would be to be able to, of course, maintain spatial resolution, have enough labeling specificity in order to be able to monitor the position of many elements at the same time. And this, of course, brings us to multiplexing. And so the way we did this was to couple the microscope to a microfluidics device. It's here appearing at the top. With the microscope, imagine we can zoom in, and I can show you here schematics of a DNA molecule. We can inject a fish probe that will target specifically one locus, shown here in red. And this appears in the microscope as a blob. We can localize the center of this blob with very high precision, so tens of nanometers. And then we can photobleach this blob and use the microfluidics to inject a second probe that now will target specifically the second locus, right? It's locus number 2 and it's blue. We can then photobleach it and inject the third one and so on. So we can repeat this process over and over again. And so at the end, we end up with the 3D coordinates of each locus with genomic specificity and with nanometer precision. In other words, what we can do in this technique is to trace the path of the chromatin fiber, and we can of course do this in single cells without complex samples such as a Drosophila embryo. So the addition of RNA fish also allows us in additional cycles of imaging to see which of these cells have a specific transcription hotspot for a gene. So we can detect which of the cells are transcribing and which of the cells are non-transcribing. And I want to say that concurrently, similar methods were developed by the lab of Schreiber-Funk and by the lab of Alistair Buttiger. And so how can we use this method in order to answer the question that I said at the beginning, whether that organization is related to transcriptional activation? So to do so, we have selected a region in chromosome 3L of the Drosophila genome. This region contains two different dots, dot number 1 and dot number 2, that you can see there in the HiC map. We imaged this locus using HiM, and I can show you here the matrix that we get from HiC on the left and from HiM on the right. You can see already that there is a very good resemblance between the two. We can visualize clearly the existence of this dot number 2 and dot number 1 in the HiM matrix. But you may be thinking, well, this is nice. However, this is still an ensemble matrix. So what about single cells? So we can, of course, represent the equivalent matrices for many cells. It's about 100 cells. And before you start zooming into your screens, you can zoom in 4 of these cells, and we can really show you any possible scenario. You can see cells like this first one on the left that has a chromosome organization that is similar to the one we see in Ensemble. However, you can see many cells that display a very different and distinct chromosome organization. So, of course, we wondered what could be the source of this heterogeneity. You may be thinking yourselves, maybe it is cell cycle, right? We know that cells can have different chromosome organizations depending on the cell cycle stage. But in this case, we're looking at Drosophila embryos. And I always try to use this excuse to show this beautiful movie taken by the lab of Punyanaga, where you can essentially see how cell division progresses during development in Drosophila. So you can see very clearly is that all of the cells go through the same cell cycle at the same time. So if we fix embryos at a specific nuclear development stage, then we know that all of the cells are in the same cell cycle stage. So the heterogeneity in this case doesn't come from cell cycle. And so, of course, we wondered whether this heterogeneity may be coming from transcriptional activation. And so our first, probably blunt way of doing this at the beginning was to measure the volumes of each of these dots independently. So, for example, we can take the locations of the barcodes, the Lossian that we imaged in the tab number 2, and from this we can calculate the volume of this dot. Of course, I told you we are imaging this in the context of a Drosophila embryo, and we have also array fish data. So I can tell you that the stop gene that is in the tab number 2 is expressed in these cells in blue at the top, that is not expressed in these cells in pink in the bottom. Right? So you can, of course, derive volumes of dots where this gene is expressed and volumes of dots where this gene is not expressed. So the way of representing this data is as follows. I show you here again the part of the Drosophila embryo. Each of these dots here is a different cell, and the color represents the size of that gene. Right? So if you zoom in, you see straight away that there is a very high degree of heterogeneity. You have many cells where this dot is very small, and many cells where this dot is very large. If we now show you side by side the expression pattern of this DOC1 gene, you see that doesn't seem to be any apparent correlation. We can get more quantitative analysis by just measuring the range of duration for the cells where DOC1 is off, and the same for where DOC1 is on, and we don't see any evident difference. So, conclusion from this analysis is that that shape alone does not seem to determine function. Next, we moved to a second question, which is maybe what we can do is try to see if the presence of a border between these two dots is affected by expression of DOC1, or seen from that way around. Does the expression of DOC1 is the expression of DOC1 influenced by the presence of a border? So to do this, what we did at the beginning was to represent the data again in sample metrics. We have calculated the mean insulation score for this matrix, and you see these two bumps here. The second bump essentially corresponds to the border between dot number 1 and dot number 2. But of course, you may be thinking this is a bit useless because this is really data from the ensemble. We can do the same in single cells. And the way this is represented here is that each row represents a different cell, and the color code here represents the insulation score. So yellow represents very high insulation scores, so the two dots are very segregated. Blue represents very low insulation score, that means these two dots are probably fused. We can, of course, also represent the transcription stages. Here, red means on, white means off. And if I zoom into this region at the top contains the cells with the highest insulation scores. You see that even cells with very high insulation scores can be either expressing or even not expressing the DOC gene. So there doesn't seem to be, again, a correlation between gene expression of DOC and the existence of a TAT border. We can look at the other end of this map, and here we see very low insulation scores. It means here that probably the two TATs are very close together. There is no border, physical border between the two. And again, you see many cells where DOC1 is happily expressing and many cells where it's not expressing. So again, the conclusion from this is that borders are not strongly correlated to transcriptional status in this space. And so to summarize briefly, I have told you that we detected a very high degree of heterogeneity in single cells. We have analyzed single cell volumes and single cell borders, and we did not find a correlation between the existence of different sizes of TATs or the presence of borders with transcriptional activation. And so, of course, we wondered, maybe the information that is important is in the specific interactions between functional elements within TATs, so intra-TAT interactions. And so to start looking into this, what we did first is to go back to our contact metrics that you've seen now several times, TAT number 2 and TAT number 1. We can select an anchor, and we can ask the question, with which locus is this anchor interacting in single cells? So every time we see an interaction with locus 1, we draw an arc. When we see an interaction with locus A, we draw another arc. And so if we represent this data from single cells, these arcs essentially accumulate. The higher this curve, the more cells that display this interaction. And so this already is telling you that this anchor, specifically in TAT number 2, is seeing 70% of the time loci that are in TAT number 2, and only 30% of the time loci that are in TAT number 1. So, of course, this means that these loci within the TATs prefer to interact with loci in the center. Right? But you probably are wondering yourselves, where are these peaks? Where are the function elements of these peaks? And where are the function elements in this angle? And so, in order to answer this question, I need to explain a bit more about the biology of this TAT number 2. This TAT number 2, we call it DOC-TAT, because an obvious reason it contains 3 genes, DOC1, 2, and 3. And contains also several enhancers. I'm going to focus on these 4 enhancers here, EA to ED. Right? And the question, of course, is, can we detect the interactions between these cis regulatory elements, and can we correlate these interactions to the presence of a transcript? So to do this, we need to do 2 things. First, of course, as before, we need to concentrate only on the cells that are expressing DOC. So for this, we look at the pattern of expression of DOC, and we select only these cells. And we need to increase the resolution of high-M of this map by about 3-fold in order to be able to have enough resolution to differentiate these regulatory elements. And so when we do these 2 things, we get the matrix for DOC2 for the DOC-TAT that I've shown you before, and this looks like this. You can see straight away that there are dots. These dots remind us of the dots that people have seen in Mendelian high-C maps between TAT borders. And so what they represent, of course, is specific looping interactions. So what are the elements that are involved in these interactions? The best way of looking into this is again by selecting an anchor and now drawing the profile of interaction of this anchor with all of the other barcodes. Right? When we do this, we essentially have a virtual 4M profile. Again, the peak here in A and B means that this anchor preferentially interacts with A and with B. And so we can do this analysis by selecting the enhancer C as an anchor. When we do so, we see that the maxima appear specifically at the positions of the other enhancers. So that means that this enhancer C preferentially interacts with the 3 other enhancers on this locus. We can play the same game, but now selecting the DOC1 as an anchor, so the promoter of this gene. And what we see is that this gene tends to interact with the 4 enhancers in this locus. So conclusion from this slide is that this technique allows us then to detect enhancer-enhancer and enhancer-promoter contacts in cells where transcription is activated. And so, of course, we wondered whether these cis-regulatory interactions were widespread. So for this, we can repeat the same 4M analysis, but now without enhancers. When we do so, we have essentially the same answer. So if we select, for example, enhancer A, we see that they interact preferentially with the other 3 enhancers in the locus, and the same result we see for the other 2 enhancers, right? So enhancers tend to interact together. To have, let's say, independent method of analysis of this, what we did was to use Shrek 3D. So this is a method that was developed by Julien Mosiconache, and that allow us to obtain the topological representation of the high-end map, right? So remember, this is not a structure, this is a topological representation. And what this topological representation shows is that enhancers tend to be at the center of the tab and seemingly making a cluster. Of course, you were wondering, this is a good conclusion, but from ensemble data, do we see enhancer clusters in single cells? So in order to answer this question, what we need to do is to do what is called a 3-way interaction. And this relies on the following. We can select again an anchor, in this case, imagining the enhancer C, and we can look at how frequently this enhancer C interacts with 2 other loci at the same time, right? And so by making a map now of this, of course, in these maps, the anchor itself is removed from the matrix, so appears in blue. And again, we see these dots. And these dots would represent is that at this loci, we have 3-way interactions between the anchor plus 2 other loci, right? And when we see and look at the identity of these spots, we see that systematically it contains enhancers, right? We can do the same analysis for the other 3 enhancers in this locus, and we see always the same, right? So any anchor we select as an enhancer, what we see is preferential interactions involving 3 elements that systematically contain enhancers. And so this makes us think that maybe enhancers here, they exist in single cells, and promoters need to actually sync into these clusters in order to get activated. So to test this hypothesis, what we did is to do the same analysis, but now selecting the promoters as anchors, right? And what we see now is that these promoters tend to interact with lower efficiency, but they tend to interact specifically with the enhancers. So what this is again telling us is that most likely we have preformed enhancer clusters, and promoters are from time to time interacting with these enhancer clusters to get activated. Right? So promoters contact enhancer clusters in single cells. And so, of course, we have been looking so far only at cells where the DOC gene is actively expressed, right? And we imagined that we would not be able to see these interactions in cells where this was not the case, right? And so before I move on, I just want to remind you of the biological system. I told you that these DOC genes are expressed in a specific pattern of the chromosome embryo. This nucleus cycle, it is what is called dorsal ectoderm, so it's a presumptive dorsal ectoderm of the embryo. There is 2 other tissues, the neuroectoderm, here shown in orange, and the mesoderm, shown in green, where not only the DOC genes are not expressed, but their expression is actually repressed, because if they express in these other tissues, this will lead to developmental defects. And so what we can do is calculate the high metrics in this tissue and compare it to the high metrics in these other tissues where this expression of this gene is repressed. And the surprising result is that the matrices look amazingly the same. So the upper right triangle is the matrix we have been seeing so far. So this is DOC1 RNA. So this is the cells that are expressed in DOC1. And the mesoderm, the matrix is essentially cells that are expressing snail. Snail is a marker for the mesoderm. What you see straight away is that the matrices look really similar. If we do 4M analysis, selecting different anchors, we see exactly the same. So the profiles of interaction are amazingly similar. So what this means is that these enhancer clusters are also existing in cells where the transcription of DOC1 is repressed. And again, promoter interactions with these enhancer clusters also appear to happen in these cells. So of course you may be wondering, and we wonder for a long while, why this may be the case, right? Why are these specific interactions also happening in off cells? So to try to get an inkling into this, what we've done was to look at what factors are specifically binding to the dorsal ectoderm, where DOC is expressed, and to the mesoderm, where DOC is repressed. So when we look at the first, we see notably, these are the three enhancers I've been talking to you about, underlined here in red, in green. And if we focus on these two elements, MAT and SEN, these are transcriptional activators. So this makes sense. These enhancers are bound by transcriptional activators in the dorsal ectoderm. So when we have enhancer promoter contacts there, they will tend to activate the transcription of the DOC genes. But the surprise comes when we look at the binding pattern of transcriptional repressors. Right? And what we see is that these enhancers in these other tissues are actually bound by these repressors. And so what this means here now is that when promoters contact enhancers, now they will be essentially contacting transcriptional repressors. Yeah? And so the model is this, is we have two different tissues, the dorsal ectoderm and the mesoderm. In the dorsal ectoderm, we have enhancer hubs, we have promoters, and sink into these enhancer hubs, and because these enhancer hubs are populated by active transcription factors, then of course these will lead to the activation of transcription of the DOC genes. In the other tissues, these enhancer elements are still forming hubs, but now they're occupied by transcriptional repressors. And so what it means is that when the promoter gets in contact with this repressive hub, what it does is essentially enhance the repression of genes. Right? So in this sense, chromatin architecture can play a double role in transcriptional regulation. And so of course at this stage we wondered whether this network of cis-regulatory interactions appeared before DATs emerged or before the onset of regulation of gene expression. And so another way of asking this question is to ask whether these interactions appear before or after the emergence of DATs. Right? And so we looked at this in three different slides. First, we want to look at when are these cis-regulatory interactions established, right? I remind you, we are looking at different stages of embryogenesis in Drosophila. And I remind you as well that the onset of cytotoxic transcription happens in these very last 4 nuclear cycles from 11 to 14. So far, I have been showing you data for nuclear cycle 14, where I've shown you this matrix of interactions. We can do the matrices for previous nuclear cycles, and we see very similar matrices, right? So that means that these cis-regulatory interactions really appear very early in embryogenesis, as early as nuclear cycle 11. So of course the question is, are the DATs established as early as nuclear cycle 11, right? And so to look into this, what we did is again a Haim experiment in this circuit, but now not only in the DOC-DAT, but in a larger region encompassing DAT number 1 and DAT number 2, so we could see the border. You have seen this data already, and I told you already that in nuclear cycle 14, this DOC-DAT is formed. When we do the same experiments, but now in previous nuclear cycles, you see that this border doesn't exist anymore. So the DOC-DAT is not formed in these previous cycles, right? This is concordant with the high-C data of the lab of Juan Navacaristas and Giacomo Cavani. And what it shows is that these cis-regulatory networks precede the establishment of DOC-DATs, of the DOC-DAT at nuclear cycle 14. And so the final question is whether these cis-regulatory networks appear before transcriptional activation or not. And so in order to do this, what we do is we do imaging of transcription at different developmental stages. You can see the transcriptional hotspot as a green spot in these images in nuclear cycle 14. So we know that DOC is expressed in this nuclear cycle. If we look at previous nuclear cycles, we see that this gene is actually not expressed in these previous cycles. And we can also look at RNA-seq data from the literature. And what we clearly see also in this case is that none of the DOC genes is expressed before nuclear cycle 14. And so from this, we can conclude that, again, these cis-regulatory interactions are established very early, nuclear cycle 11, and this is 3 nuclear cycles before even the activation of the DOC genes starts. So this formation of these networks is prior to transcriptional activation. So I get to the take-home messages slide. I have shown you that we have developed a technology that allows us to detect cis-regulatory logics and transcriptional state in single cells. with spatial resolution. We have used this technology in order to see that we observe cis-regulatory hubs with both activators and repressor elements. We have hypothesized that this may mean that chromosome architecture can play a double role in activating transcription, but also in ensuring repression of transcription in some cases. I've also shown you that this network of cis-regulatory interaction precedes the emergence of TAS or the onset of transcriptional activation. And going to pacification, without mentioning that similar biological results were also obtained by the lab of Juan Vacarizas, and probably he's going to be talking about this tomorrow in his talk. And so I want to end by the first or second slide that I've shown you today. I told you that many instrumental or developments were required in order to discover new levels of chromosome organization. I've told you that genome-wide technologies have enabled recently for the discovery and for the characterization of TADS, hubs, compartments, many new levels between the nucleosomes and the chromosome territorial levels. Single-cell imaging technologies have shown very recently that these levels of organization are highly heterogeneous. I could say that so far it's unclear how and whether and if 3D structure encodes function and whether this depends on the system we're looking at. And I think it will be critical in the future to use interdisciplinary methods in order to be able to get a full understanding of these structural levels and the functional roles. And so I think it's very timely to have 3D genomics meetings today. So without further ado, I will end by thanking the people in my lab that have done the work that I showed you today, particularly Sergio Spinola and Marcus Goetz. I also want to acknowledge the great collaborations with the lab of Giacomo Cavalli and with Munia Laga, and also funding sources for their continuous support. And I'm going to end by asking you a favor. If you point your mobile devices to this QR code, this will take you to a survey where you will be able to tell me if you understood the talk, what part you didn't understand, where you were lost, and if you were lost or confused by any reference. Thank you.
So does the high-C data of the TADS mentioned here also look different in active and silent states? The last part of your talk.
No. This is a brief answer. I think Juanma Vacarizas will tell you probably more about this tomorrow. I don't want to preempt his message. But the two data sets are important.
So we have also another question from Ivan. So if understood, high-M is more specific than high-C. So does it mean that the effective radius of contact is smaller? Because do we know the radius of contact of IC, maybe? Because you are controlling the radius of contact, but...
Yeah, this is a very good question. I don't know. I mean, I think that maybe, the radius of contact at IC, most people think is in the order of 150 to 100 nanometers. This region, it's difficult to know, whether this depends on the cross-linking conditions or the sequencing method. In our case, as you say, we control what we call the contact frequency. Most I can tell you is that we get good agreement when we use small contact ratio, right? So like 150, 100, 150 to 100 nanometers. But this depends also on the genomic scale, right? This example, I'm just showing you a very small locus, right? If we look at barcodes that extend over the whole genome, so they're much more spread, then we find that the best distance to get the highest correlation with high-C data is more like 400 to 500 nanometers.
So we have another question from Vipin. So how far away can these enhancer cluster activate promoters And would this property vary depending on the activating factors bound to them?
I mean, the short answer is that I don't know, because we haven't done the experiment of displacing these enhancer elements farther away than they are. Many of these enhancer elements have been characterized in trans assays, right? In these assays, what you do is you essentially generate an artificial construct where you put your enhancer, and you see that this enhancer still activates these genes, right? So I guess it can be very far away and still activate these promoters. Of course, the activation level will depend on the composition and on what transcription factors, how many are present, right? I guess the bottom line message from our study in this locus is that, the textbook image of an enhancer alone coming in contact with the promoter, the context of enhancer clusters is different, right? Because there you have many enhancers with similar transcription factors clustering together, and what you will have instead is a promoter really sinking into this pool of enhancers, right? And so perhaps even if you take one enhancer and put it very far away, if this enhancer manages to get into this enhancer hub, then you still have activation.
Okay, so we have a question from John. John, you can ask your question if you want.
Yeah, so I got the message that there's a substantial effect on gene regulation by establishment of these cis-RE type interactions before the TAD in that case that you analyzed appeared. I'm always wondering what fraction of activation depends on sort of these initial just molecular interaction events inside the TAD area, and how much actually depends on formation of a nice square TAD. Do you have any kind of general quantitative rule for telling us how to think about that?
So in this, what we have observed is that the formation of these regulatory interactions precedes the formation of TADs. So in previous nuclear cycles, we don't really have TADs. We still see this cis-regulatory interaction. But of course, it happens in our case that we also don't have expression, right?
Okay.
And so in this case, we cannot disentangle the formation of TADs with the formation of these cis-regulatory interactions. An analysis that I have shown is that if we look at the nuclear cycle where these genes are expressed and where TADs are already formed, and we try to correlate expression with the presence of a border, we see very weak correlation.
Yes, I saw that, yeah. Okay.
So, okay, we have another question from Francesca Palladino. Francesca, do you have a question? Your hand is up? No, the hand is down, no. So we have another question from Olivier. So Olivier Cuvier. So he said, at the scale of a TAD, can active CRMs, I don't know what CRMs means, and repressive ones be intercalated, or do they function by competing with one another? Chromatin regulators, maybe.
So I think he means cis-regulatory modules.
Yes, maybe, yeah.
So it's another way of saying cis-regulatory elements. So what we see very often is that there is a systematic three-way interaction between two enhancers and a promoter, right? Would suggest that you don't have one or the other, right? You don't have one enhancer contacting the promoter at any given time, right?
Okay. So we have another question from Spark. So he say, if the experiment was time resolved, how long do enhancer or enhancer promoter contact last? But I think it's not live microscopy.
Yes, yeah, sure. This is fixed microscopy, so we have no time resolved information. It would be amazing if we could do this in time. People are now being able to detect enhancer promoter interactions in live imaging experiments. However, they can only look at a discrete number of 2 or 3 elements at any given time, right? So what they see is that the contacts between enhancers and promoters are very transient, right? And that enhancers can contact promoters for the initiation of transcription, but that these contacts do not need to be maintained for for the transcriptional phase itself, right, for the elongation phase. And so I guess one thing that one misses in these experiments, of course, we can measure the enhancer-promoter interactions and they're very transient. However, we can only look at one enhancer at a time. So, it would be beautiful if we can have dynamics where we look at several enhancers that can activate a single promoter at the same time and see whether a promoter, for example, is contacting any given time a different enhancer, right? And this, I guess, is we cannot do it.
Okay, yeah, maybe Luca tomorrow will give part of the answer of that. So we have a question from Julie Seguini. Have you studied any silencer instead of enhancer? And do you think they form the same kind of cluster?
So I think, yes, I think the answer is already in the presentation, right? I think people assume most of the time enhancers are activators, right? What we see is that what we have defined as an enhancer actually can act as an enhancer, as an activator in some tissue, and can activate as a repressor in another tissue. So it really depends on what transcription factors are bound to this enhancer.
Okay, and maybe to follow this, there is a question from Francesca Mugianesi, who asks, what is the physical nature of these hubs? Is it linked to phase separation or oligomerization?
We have no idea.
Okay, no clue.
Sorry, this it's something that we cannot really speculate because we need experiments in order to support either one of the models. Right?
So another question from Stefan Zayen. Is it known if the disruption of one or more of the enhancer cluster influence the expression levels of the gene, of the dog gene, actually?
So, no, it is not known in this case. We have currently doing experiments where we are removing one of the enhancers, right, to see what is the effect in the formation of the enhancer clusters and what is the effect on the transcriptional regulation of the genes that are located nearby. But I unfortunately cannot give you the answer because we don't really have the results.
Okay, thank you. So we have maybe two last questions. One from Mr. Kurukuti, I think, or Mrs. I'm not sure. So what is the influence of hybridization of the probes on your measure? I mean, do you have any idea on the TAT volume, for example, or on the distance you measure?
This is very difficult to assess, right? Of course, I mean, the hybridization protocol, the FISH protocol, we'll know is disruptive and can disrupt chromosome organization at the short scales, right, the nanometer scales. So far, what we can say is that there has been a very high degree of correlation between the high-C data and the high-M or chromosome tracing data that people have obtained, suggesting that the kind of disturbance that you get in chromosome organization by doing hybridization does not appear at these kilobase scales, right?
And so the last question would be, do you know whether the enhancer clusters also show transcription activity? So do you have evidence of production of enhancer RNAs, for example?
Yeah, this is a very nice question. I have no idea. But it would be great to be able to address it. Of course, this can be addressed by doing RNA FISH at the same time as chromosome conformation.
Okay, good. So thanks again, Marcello, for this great talk, for the lively discussion.